All right, guys. How you doing?
Good, how are you?
Good. So, and I told this morning's group this too. The university has sort of indicated that, like, when we have classes that are run exclusively Zoom, and I'd appreciate this anyways, that they want students to have their video on. It's so nice to be able to see people too, rather than just talking to your names. Andrew at least has a picture up, but if I can see your faces, that's always so much nicer for me. So thank you. Um, so what we're going to do today is it's gonna we'll probably work for about an hour, and then we'll take maybe like a 15-minute break, and then we'll come back and kind of do a part two. So, normally what I've done in this lab is it is a little bit of wet lab stuff, but it's more me demoing some wet lab stuff. But I'm going to just kind of try to walk through it because I didn't want to have another lab that we had to split up. So I think it worked well this morning with what we did to kind of try to go through and kind of brainstorm how an FPLC works. We're going to have sort of a demo protein mix that we're going to purify, or at least I'm going to tell you how it gets purified, and then we'll kind of have you guys analyze the results from that. And then we're going to work in the last part on a simulation. So for better for worse, a lot of what today is going to be is a bit of just me telling you about stuff, note taking and all kinds of stuff like that. But I think this morning's group, when they left, felt like I think I understood how proteins get purified, because that's what we're doing over the next 2 weeks. And that's kind of why I decided that we really needed to have this lab, even though it was a virtual lab, because it'll make a lot more sense for you guys next week if you actually have sort of gone through this lab first. So part of what we're going to do here is go through and answer the pre-lab questions here. So some of these pre-lab questions are going to lead us into understanding how some things work. So the first thing I want to do is just take pre-lab question 1, and I'm going to go through these 5 proteins here. So with some of them, I'm just going to kind of tell you a little story about them. These are all proteins that we're going to, with the exception of papain, these are all proteins that we're going to study or learn about in some way, shape, or form. So just some little stories that go along with them. So the first one is glucose oxidase. Why this is actually really interesting is this is an important enzyme used in glucose monitors. So when you think about how a glucose monitor works, and I don't know if anyone has ever sort of tried to test their own blood sugar. You got to prick your finger a little bit, and then you kind of take a little bit of blood out on a little piece of paper, and then you put it into an instrument. Has anyone ever told you what happens in that instrument? So it's actually the enzyme glucose oxidase. And what this enzyme does is it does oxidation chemistry. What do you guys know happens with oxidation chemistry? Oxidation is the what of electrons?
Loss.
Loss of electrons. So what happens is you actually lose electrons. Do you guys know what electricity in your walls is? That's also electrons. It's electrons in your wall. So what happens is we get glucose to lose electrons. We create a little circuit, and the flow of electrons through that circuit is directly proportional to how much glucose is there. So neat little chemistry that basically relates the number of the number of electrons that flow through a wire with the amount of glucose that's there. So we'll learn more when we, again, diabetes is going to be a thread that we carry sort of throughout here. So we'll talk more about glucose monitors a little bit later. But someone want to share what was the information that you found out about glucose oxidase? I need to know a molecular weight and then a PI.
My molecular weight was weird.
What was it?
It was 160 kDa.
Mm-hmm. Yep.
Okay, so that's not wrong.
No. So
that
stands for kilodaltons. So a dalton is essentially the same thing or related to grams per mole. So you normally think of molecular weight for regular things like grams per mole, but we think about it in terms of a unit called daltons for proteins. So proteins tend to be on the order of at least 5 kilodaltons. You know, the ones we're going to see here, this is the largest one, or one of the largest ones we'll see at 160 kilodaltons. How about Corinne for a PI? What did you get as a PI for that?
I got, excuse me, sorry. I got 4.2.
Yep. So one of the things that we're going to note here is I've also intentionally chosen these 5 proteins because their molecular weights are, some of them are similar, but some of them are different, and the same thing with their PIs. So sort of a big thing that we'll talk about here when we do protein purification is how do I separate what I want, my protein, from all the contaminating proteins. So I'm going to need to find something that's different or unique about my protein in order to do that. So I don't have really an interesting story to tell about papain. It is a protein digestive enzyme. It's not one we're really going to study, but it kind of fit into this group of 5 proteins that we were going to study here. How about a molecular weight for that? How about you, Andrew? What did you have as a molecular weight for papain and a PI? Can't hear you yet. All right, let me go to Emma. You are the next in my list of people. What did you have for a size and a PI for papain?
For molecular weight, I had 23,406 Daltons.
Yep, which is 23.4 kilodaltons. And how about a PI?
I had like 2 different values. One was 8.75, and then the other one was 9.55.
Okay, so let's say it was pretty much between 8.7 and 9.5. Okay. Esterase. So esterase, a lot of these enzymes we're going to find have digestive sort of components to them. So the little story that goes along with this is this form, or it's going to break ester linkages in triglycerides. And let's see, Lexi, what did you find out for esterase?
31,463 Daltons, and then the PI was 6.54.
And I think this morning's group had a little bit different. So what did you have is the molecular weight?
31,463 Daltons.
Okay, so let's just say 31.4 kilodaltons. Now, one of the things we found out this morning as we were going through these answers is it can be species specific, because someone found an esterase that was like 100 and almost 170 kilodaltons. So we can have a lot of variability here, but for this little exercise, it's not gonna matter too much. Carbonic anhydrase. Is that one we've heard of before?
Yeah.
Let's see, Connor. What did you get for carbonic anhydrase?
I got 30 kDa.
Mm-hmm.
Not sure if that was right, but then I
Yep.
couldn't find a PI for it. The closest one I don't know if it was right. It was 5.9.
Okay. How about anybody else? Did anyone else find a KD or a PI for that?
I had
I did 6.6.
6.6. All right. I'm going to put 6.4 to 6.6, which is in line kind of with what people, but again, this is going to be specific as to what organism you might find it from. And just reminding everybody, right, we know carbonic anhydrase is important for kind of managing the disguise of carbon dioxide as bicarbonate. And then we've got cytochrome C. Has anyone heard of cytochrome C before? Know where we might find it? This is a really critical component of the electron transport chain. So we're going to learn about it later. But one of the things that when I do this lab in person, I have little vials that show each of these 5 individual proteins, and I ask students to notice, did you see anything unique about them? One of the things that they'll notice is that, and I'm going to do this here, glucose oxidase is yellow. So we're going to see some color that we have to these proteins. And then cytochrome C is a red, like a brick red color. And what's interesting with cytochrome C is it has that red color because it also has a heme that's bound to iron. Oh, you guys are not even seeing. You need to tell me. I'm writing all this stuff. Say, Dr. Prenny, we can't see what you're writing. Oh, hang on a second. I'm not even connected with my iPad here. I'm just connected on there. So hang on a second, then I'm going to give you a minute to write. Darn it all. You guys got to speak up sooner when you're like, do you think you want to be sharing your screen with us? Sorry about that. Had to run to put out the fire in the kitchen. Now, he really didn't start a fire, but he wasn't supposed to start macaroni and cheese on his own. I went down when we had a break with the first lab at noon. I went down. I said, anybody hungry? I said, I've got 15 minutes that I can make you lunch. Nope, not hungry yet. I said, okay, you're going to be hungry later. And then he comes upstairs like right before lab starts. And he's like, I started making macaroni and cheese. I'm like, no, you're not supposed to do that. Not on your own. All right. So you probably have a lot of this information. So I don't think I need to like repeat it, but you can just kind of see all of this information here that we've been talking about. So glad I realized this before we got too far along here. So cytochrome C has this red color because of a heme that's bound to iron there, but this is actually doing a different kind of process than we saw with the heme and hemoglobin. That was for the reversible binding of oxygen. In this case, the heme and the hemoglobin that are here actually do undergo an oxidation chemistry. So we'll learn about this later, but that's kind of an interesting thing that this iron does go to an iron 3+, and that's how it transports electrons in the electron transport chain. So it actually does undergo that oxidation chemistry. But importantly, we need some data on this. So who have I not heard from yet? I think Kate, I haven't heard from you yet. What did you find out for cytochrome C?
I had molecular weight of 11.7 kilodaltons, and then I had a PI of 9.6.
Good. So what we can see here, and the intent of this initial exercise was I've got a range of molecular weights. We kind of have about two, three sort of categories here. Again, if esterase happened to have fallen in a very large category, we'd have, these very small proteins like cytochrome C and maybe papain and carbonic anhydrase are a little on the smaller side, maybe medium-sized protein. And then we've got kind of a large one with glucose oxidase. And then we also have a range of PI values. So kind of ranging from something quite low with glucose oxidase to something on the high end here with cytochrome C. Okay, so we're going to come back and revisit this slide because it will be important for us to think about how we can use this information to rationalize a separation of these proteins. So again, remember in lab, imagine I had shown you little vials with all 5 of these proteins. We kind of talked about them. You saw some color differences and so forth. And then what I physically do is I mix them all together. And then we use our FPLC instrument to purify them. Okay. And while the instrument's running, I usually go through and we write a little scheme for how this machine works to separate proteins. And let's see, I started at my last slide here, so I'm going to move this.